Good morning, dear students. Today we are going to learn about the database and the source of database like primary and secondary databases and how these databases can be used for different purposes in bioinformatics. Before that, we should also understand that there is one more application of bioinformatics, that is drug discovery. Using computational tools, the cause of disease is identified and treatment is given with appropriate drugs. Okay, so you must have seen these days new drugs are coming in the market, and they have biotechnology as the base or molecular biology as the base. So these new drugs for different diseases can be discovered with the help of bioinformatic tools. Okay, the causes of disease is identified and treatment is given with appropriate drugs. So they are more appropriate than earlier research because they have been proved for the treatment for that particular disease. Now next we are going to learn about the databases. Okay, the databases tools and their uses. Databases, in the most general sense, it is an organized collection of data. Okay, so while whatever biological data is there, which is available in the form of maybe in the form of proteins, maybe in the form of nucleotides, genes, that data is get stored. It's a lot of data, huge data, and through manually manually you cannot solve these problems, okay, to understand or interpret the data. So this data which in different places scientists are working on in the field of biotechnology, molecular biology, in proteomics, genomics, they submit these research papers to NCBI. NCBI stores it in different databases according to, they categorize it and they put it in different databases, and that database is now available for the use of all the scientists and research community. Okay, it is an electronic system that allows data to be easily accessed and manipulated and updated. Okay, for example, if I have discovered some new gene sequence in jamun or in mango, I have to submit it to NCBI. NCBI check whether it is a new gene sequence or earlier somebody has reported it. Okay, then accordingly they will put that gene sequence in your name in some database. Suppose some X person want to know about that, he can easily instead of asking to me or searching me, he can easily go to NCBI website and he can search for that data, okay, or a database. It is used by an organization as a method of storing, managing, and retrieving information. Modern databases are managed using the database management system, that is DBMS. Okay, so the organization which is there, which store the data, manage the data, and retrieve the information according to the requirement. Okay, so there are different types of databases. First one is the biological databases. A biological database is a collection of data that organized so that its contents can easily be accessed, managed, and updated. The activity of preparing database can be divided into collection of data in a form which can be easily accessed. Okay, so second is making it available to multiple users. As I said, the data will be collected from multiple sources, like different scientists they are working. They will uh, write the research papers or the research articles that will be submitted to international or national level journals, which are in close contact with NCBI. NCBI take the important research papers and they put it in their database so that it can be available to multiple user system. As I said, NCBI is a working like a library, storing the information and providing it to the users. Database in general can be classified into primary, secondary, and composite databases. A primary database contains information of the sequences or structure alone. Okay, so the primary database contains the information of sequences or structure alone. Example of this include Swissport PIR of protein sequence, gene bank, DDBJ for genome sequences, and the protein data bank for the protein structures. These are some of the examples of the primary databases. Okay. Now, second, in bioinformatics, data banks are used to store and organize data. Many of these entries collect DNA and RNA sequences. Okay, from scientific papers and genome projects. Many databases are in the hands of international consortia. The major database of biological macromolecule structure is the Worldwide Protein Data Bank, okay, PDB, a joint effort of research collaboratory for structural bioinformatics in the United States. The Protein Data Bank of Europe, that is PDBE, at the European Bioinformatics Institute in the United Kingdom, and the Protein Data Bank, Protein Data Bank Japan at Osaka University, they are working under it. The homepage of the PDB partners contains link to the data files themselves, to expository and tutorial material, including news items, to facilitates to facilitize for deposition of new entries, and to specialized search software for retrieving structures. Okay, so we are going to do it in the bioinformatics practicals. So in that you can easily understand how we can search a research paper or research article based on our query or this whatever uh, information we required. We can search it by giving different by using the search engine in from the NCBI, and then that research paper we can use for our own research work. Information retrieved from the data archives utilizes standard tools for identification of data items by keywords. So it is not only that they have stored scientific information, but they have stored general information about any scientific topics. Okay, related with biology, when the fishery, aquaculture, everything is available. Almost everything is available in the NCBI, so that you can use it as in the format of notes also. There are some research paper, there are some good research articles which can be used to get information about particular topic, that is also available in NCBI. Now, what is primary databases? In bioinformatics, databases are categorized as primary or secondary. Experimental results are submitted directly into the database by researchers, and the data are essentially archived in nature. Once a database accession number is given, the data in primary database are never changed. They become scientific record. Okay. So in last year, we have studied how to do research work, how to write a research publication, different types of research publications. So whenever you are doing some new research work, you have to go in the that first of all you have to find out the problem, then you have to work on that problem find out the research methodology. Then you have to do the research work, find out the results, that is your observations. Then finally conclusion, and then you'll write a research paper accordingly, and then you can check it for plagiarism. Then you will submit it to the scientific journals, reputed scientific journals. Now UGC has also given some list of scientific research papers, which is called as UGC care list, and in that only you can submit papers, because some journals are fake journals for collecting money. They are uh, giving different attractions to the researchers that it will be published very fast. You have to just pay 2000 3000 rupees. But these journals are fake journals. They are not taking any kind of review of your research work, and even they don't check even plagiarism. So these journals we should avoid these journals. Okay. So the most common use of bioinformatics for biologists is search within primary databases, which include sequences and three-dimensional structure databases such as protein-protein interactions, large-scale mass spectrometry analysis, and identification of lipids, sugars, or small molecules metabolites. Okay, so you can see it here. Like this, you can study. You'll get the search about the different topics in the primary databases. Okay, so you can easily study about the protein-protein interactions, then data of large-scale mass spectroscopy, and even you can identify the lipids, sugars, or small molecules metabolites. Next, we can see about the database of nucleotide sequence was launched at NCBI in 1992 in collaboration with European Molecular Biology Laboratory, that is EMBL, and the DNA Data Bank of Japan, that is DDBJ. You have to remember these long forms, okay, short forms and their long forms. So European Molecular Biology Laboratory, that is EMBL, and DNA Data Bank of Japan, that is DDBJ. The roots of European Bioinformatics Institute, that is EBI, lie in the European Molecular Laboratory (EMBL) Nucleotide Sequence Data Library, which was established in 1980s at the EMBL in uh, Heidelberg, Germany. This was the world's first nucleotide sequence database, and the EBI was established on the Wellcome Trust Genome Campus in the United Kingdom in close proximity to the Wellcome Trust Sanger Institute of the Human Genome Mapping Project Resource Center. The EBI fulfills the three important functions. First one, it provides some of the world's largest and the most heavily used bioinformatics services in the free and unrestricted manner. It carries out research and it trains the bioinformatics of the future. As I told you, this particular topic has a lot of scope, though in India it has comparatively less scope in small cities, but in cities like Pune, Mumbai, Bangalore, Chennai, where they are carrying the work on bioinformatics, there's a really lot of scope. You can do after MSc, you can go for a small course, 1 year or 2 year course in PG diploma in bioinformatics, and there are many jobs available. Those who have missed to become the software engineers, or if you have a good liking in computer or softwares, then you can easily now also, after completing zoology, you can go for bioinformatics. In foreign countries, there's a lot of scope. If you'll search on the Google for jobs in bioinformatics, you'll get a lot of openings. It is a DNA sequence database maintained by the NCBI Bank. Currently contains 150 billion base pairs of information in 160 million sequence files. Only original experimentally derived sequences can be submitted to Gene Bank, and it is a redundant database. A particular sequence can be determined by independent research projects, cloning of the single gene, or by genome sequencing projects. GenBank continues to grow at the exponential rate, doubling every 18 months. Presently, the major sources of submitted sequences are genome projects. Okay, so completed sequences of full genetic material of organisms. As I said, the sequencer can be used for sequencing of genome, and that information will be shared with bioinformatics tools, so that it will get arranged in proper manner, and the data will be sent to the NCBI databases. Up to now, more than 1,000 genomes have been sequenced, including Human Genome Project, which was finished in 2003. The human genome sequence and in fact, most of the genome sequences are freely available in GenBank and in other databases, for example, Ensemble and GeneCard. So it is an annotated database, GenBank, and the sequences are supplemented with explanation on its information content, including the coding regions of the source of the sequence and the related publications. Nucleic acid sequences and any analysis derived from the sequences can be published only after they have been deposited in a freely accessible databases. Okay, so all are freely available. There are two types of research articles. Some of them you have to pay in dollars to purchase that article, and some are open access or freely accessible databases or research articles. Newly determined nucleotide sequences can be identified by and compare using the GenBank databases using the BLAST program, and the result of this analysis are GenBank files identified by an accession code. Okay, so we'll stop it here. In the next lecture, we are going to see about the UniProt. Okay, thank you.